2009 marked the bicentennial of Darwin's birth and the 150th anniversary of his landmark book, On the Origin of Species. Around the globe, supporters of Darwin planned a year-long celebration. In Germany, one of the largest Darwin anniversary events took place in the city of Stuttgart at its State Museum of Natural History. The exhibition was directed by German paleontologist Günter Beckley, one of the museum's curators.
We had about 100,000 visitors, and the complete exhibition, together with a program that was accompanying this exhibition, was one of the largest, if not the largest event in the course of the Darwin Year celebrations in Germany.
Beckley decided to use the exhibition not just to celebrate Darwin's theory, but to make clear to the public that there was no debate about Darwin's ideas among scientists. In order to refute the growing idea of intelligent design, Beckley decided to include a display on the bacterial flagellum.
The reason why we selected the flagellum as the poster child to basically expose intelligent design was that the bacterial flagellum has a kind of iconic status. We built a mobile model of the bacterial flagellum and had this animation to show that it could originate naturalistically.
The exhibit highlighted the type 3 secretion needle complex as an explanation for how the flagellum could have evolved. In addition to an exhibit about the flagellum, Beckley came up with a display to dramatize for visitors the overwhelming scientific evidence for Darwin's theory. It was a balance with books on it.
And the plan was, on one side of the balance, we would have all the books against evolution, books by creationists, intelligent design proponents. And on the other side of the balance, we would have one book, The Origin of Species, but the balance goes down on the side of the one book because this is the real heavy evidence.
But the display didn't have quite the result Beckley intended.
And I made one big mistake. I read the books on the lightweighted side, the apparently lightweighted side. And what I recognized to my surprise is that the arguments I found in those books were totally different from what I heard either from colleagues or when you watch YouTube videos where the discussion is around intelligent design versus neo-Darwinian evolution. And I had the impression on one side that those people are mistreated, their position is misrepresented, and on the other hand that these arguments are not really receiving an appropriate response and they have merit.
One of the books Beckley read was Darwin's Black Box.
I read Darwin's Black Box, where he basically introduces this concept of irreducible complexity.
Beckley soon realized that proposed Darwinian explanations for the origin of the flagellum didn't work. The type 3 secretion needle complex was no help because it probably developed after the flagellum.
This is a reduced flagellum motor and not a precursor of a flagellum motor.
In addition, the suggestion that natural selection could have gradually built a flagellum by co-opting parts from other systems didn't make sense.
It is graphically convincing, but if you know the ontogenesis of the flagellum motor, then it is completely ridiculous. It cannot work. You cannot build the flagellum by just adding outside of the cell wall some protein elements on it and make the flagellum longer and longer. This kind of scenario doesn't make sense in terms of the ontogenesis of the structure.
Like Behe a couple of decades earlier, Beckley began to dig deeper.
And so when I read those books on intelligent design and the books by Mike Behe and Bill Dembski and the book by Steve Meyer were not existing then. I thought there is some merit to it, and I made contact with some of the representatives of the intelligent design movement. The next thing I found out is that they are much different from what I expected. They are open-minded. They are not religious fanatics who try to push a kind of theocratic system onto society under the label of intelligent design. They are really interested. Is this neo-Darwinian story really true, or is there scientific reason to doubt it?
Beckley didn't fit the usual stereotypes of a Darwin skeptic.
So many people will think somebody who comes to doubt the neo-Darwinian process and embraces intelligent design probably was religious from the very beginning, probably is an evangelical Christian and has his axe to grind, his religious axe to grind. I came via totally different path to the views I hold now. I'm coming from a family background which is totally secular, agnostic, was not baptized, didn't join any kind of religious education, never went to church, so I was completely irreligious. Was not even interested for most of my life in philosophical or metaphysical questions. I was interested in nature, in animals, and in natural sciences.
Beckley publicly disclosed his support for intelligent design for the first time in 2015. As Beckley was thinking through his doubts about Darwin in Germany, an American scientist was conducting lab experiments that would expose just how daunting the challenges facing Darwinian explanations really are. Douglas Axe earned his PhD at Caltech. He then spent 14 years doing research in molecular biology at top labs in and around Cambridge University. Axe was skeptical of Darwinian evolution, and he wanted to find ways to actually test what it could and couldn't do. When Axe returned to America, he formed Biologic Institute to do just that. Axe doesn't work on the bacterial flagellum, but he and his colleagues, Ann Gager and Marcy Reeves, have conducted experiments that help address the feasibility of Darwinian explanations of the flagellum. Those explanations depend on the idea that natural selection can easily reuse existing parts and adapt them for new functions. But Axe's experiments raise two problems for this and other Darwinian claims.
I refer to these as the big problem and the little problem. One is the big problem of inventing an entirely new protein structure to do something new. And the smaller problem is tweaking an existing structure so that it does something slightly different. Clearly, one of these is more challenging than the other. And my work early on showed that the bigger problem was beyond the reach of evolution. You have to get too much right in order to get a new structure to form, in order for accidental causes to get these new structures. So my colleagues and I, Ann Gager in particular, started to look at the smaller problem, which is, can you take an existing folded structure and evolve a new function for it? And somewhat surprisingly, we found that even this smaller problem is too hard for Darwinian evolution, at least in the cases that we've examined. The number of changes you have to make to an existing structure to get a new function to be performed by it is beyond what you can get by random mutation, by Darwinian evolution.
Critics of Behe had suggested that natural selection could build complex new machines such as the flagellum by co-opting simpler molecular machines made up of several protein parts. But Axe's research seemed to show that co-opting even one existing protein to perform a slightly new function was beyond the reach of the Darwinian mechanism. After Dover, Behe published a new book that helped explain why. Behe showed that building many complex biological structures and even new proteins would likely require multiple coordinated mutations.
Many different biological processes will require multiple mutations before they're going to have an effect.
But according to Behe, new studies in population genetics showed that coordinated mutations were beyond the reach of natural selection in many cases.
Long and the short is that if you need more than one thing to happen at a time, the improbability of getting the correct two mutations goes up exponentially. If you need to change one particular subunit of DNA, well, there's 3 billion subunits in a mammalian cell. So the odds, if you need one particular one, that's, 1 in 3 billion. Okay, well, that sounds like a lot, but there might be billions of organisms around, or at least over time. But if you need two, that means you need 1 in 3 billion times 1 in 3 billion. It goes up exponentially. You have to multiply them together. And so you very quickly run out of probability.
Behe's analysis helped explain why a growing number of lab experiments have demonstrated the limits of the mutation natural selection mechanism. Since the late 1980s, Michigan State University biologist Richard Lenski has been running a long-term evolution experiment with E. coli bacteria. A staunch supporter of Darwinian evolution, Lenski wanted to follow a population of bacteria over time and see what new functions would evolve.
I'm a big fan of this experiment because it does not put out a model. It's not a computer model. It's not a theory. What he did was let evolution happen on its own and say, what did it do?
By 2014, Lenski and his researchers had grown over 60,000 generations of bacteria.
That's equivalent to, say, a million years in the lifespan of a large animal like us. And there have been trillions upon trillions of different bacteria that have been born and died in his flasks.
Although often cited as providing evidence for Darwin's theory, Lenski's experiments are perhaps most revealing in what they haven't produced.
We don't find a new protein with a new fold with a new function. By and large, these are deletions, insertions, rearrangements of information that's already present.
He didn't see anything like the evolution of some new complex system like the flagellum. Nothing remotely like that.
At some point, you're going to have to show that you have a gene with one function has now evolved into a gene with a different function, different protein folds, and we're still waiting.
What we see going on in the Lenski laboratory and other places too is that Darwinian processes or random processes degrade information. They do not build it. They are not putting in new information. They might tweak something here or there, and at the margins you can have differences about what you call information.
Taken together, this flood of new data has raised a powerful challenge to claims that natural selection can explain the origin of new functional genes and proteins, let alone a complex biological machine like the flagellum. At the same time, new hurdles to the Darwinian mechanism are also being raised that are specific to evolutionary accounts of the flagellum itself. For example, evidence has continued to accumulate that the type 3 secretion needle complex was not a precursor to the flagellum after all.
The flagellum definitely came first.
Mutation density studies show that genes in bacterial flagella have experienced more mutations than genes in type 3 secretion needle complexes. Genes that have experienced more mutations are generally presumed to be older.
We find flagella across all phyla of eubacteria, very deep. The mutational densities are great for bacterial flagella. We find them arranged in the same genetic arrangements on the chromosome across genera, across families. For the most part, there's been some rearrangement. It was part of the aboriginal chromosome of these organisms. In contrast, type 3 secretory systems, we find in a very narrow range of gram-negative bacteria. They've been imported, and the mutational densities are much shallower, which means they're newer structures compared to the flagella.
The supposed killer explanation against Behe has turned out to be a dud. The needle complex can't explain the evolution of the flagellum because it didn't even exist until after the flagellum. But there's more. Darwinian explanations of the flagellum assume that parts already existed that natural selection could reuse in order to build the flagellum. This may well explain some features of the flagellum, but there's a big problem. Key flagella parts are turning out to be unique. They don't appear to exist anywhere else. They are only found in the flagellum, and if they didn't exist already, natural selection wouldn't be able to use them to build something else. Even if all the necessary parts were there for natural selection to co-opt, it's now becoming evident that just having the parts alone wouldn't be sufficient. Complex structures almost always require a specific sequence of assembly. When building a house, A foundation must be laid before the frame can be put up, and the frame must be completed before the roof can be added. In a similar way, a flagellum must be built in a carefully orchestrated process that is governed by assembly instructions encoded in the DNA. Merely having the various parts of the flagellum available in the vicinity of a bacterium isn't enough.
Even if you had just floating around a type 3 secretory system or a hook protein or a rotor or a drive shaft, you've got to put these things together. And biological systems aren't put together like Lego bricks. You don't just stick them together. There's a very precise sequence of expression of these genes, these protein products, that allows the machine to be built piece by piece, somewhat like the assembly apparatus in an automobile factory. That assembly routine involving multiple different genes and regulatory proteins is itself arguably an irreducibly complex system. You remove any one of those genes or one of those regulatory proteins, the flagellum is simply not going to get built. Something will shut down the biosynthetic pathway if one of those key elements is missing. So co-option in an attempt to get around the irreducible complexity of the finished product posits a bunch of parts which could only be plausibly put together by another irreducibly complex system, so you haven't really solved the problem.
The existence of assembly instructions required to build a flagellum isn't simply a challenge to Darwinian evolution. It also provides evidence of intelligent design. Encoding the assembly instructions requires massive amounts of biological information, but where does that biological information come from?
The genome is full of information. It has literally a sequence of DNA letters which tell the machinery of the cell what to make. It's like a blueprint.
We know of a cause that can produce digital code, hierarchically organized information, integrated circuitry, and that cause is intelligence. In fact, that's the only known cause that we know of based on our uniform and repeated experience, the basis of all scientific reasoning about the past, that we do know of that can produce information, and therefore it is possible to infer from those effects information processing circuitry back to intelligence as the best causal explanation of those complex molecular machines.
More than 2 decades after the publication of Darwin's Black Box, the challenges to Darwinian explanations of molecular machines like the flagellum have become even more daunting. As a result, evolutionary biologists are scrambling to find new unguided mechanisms in order to avoid facing the alternative proposed by Behe and others, that biological systems reflect intelligent design. Michael Behe raised ideas that even now are reshaping how we understand biological systems. The man who describes himself as shy helped spark a revolution.
Well, one of the things I like about Mike Behe is that he's a very humble and unassuming guy. He's not full of himself, yet he's a player on the world stage scientifically. People all over the world know of his idea of irreducible complexity. They know of the idea of intelligent design because of his work. And it's really reframed the debate. Whether you agree or disagree, one way or another, you've got to now address the question of the origin of these irreducibly complex systems that we find in living systems, whether we're talking about the flagellar motor or the ATP synthase or the kinesin walking motor protein or the circuitry or the gene expression system or whatever it is. The biology inside the box is really complex, and it's an integrated and functional complexity that requires some kind of an explanation. And I think just by highlighting that, Mike has reframed the debate in a way that I think has changed the way people think about biology in the 21st century.
For Behe, the most satisfying part of his intellectual journey isn't getting recognition, it's opening minds.
I remember in particular a time I was talking at the University of Massachusetts at Amherst, and also on the stage and also talking, it was not really a debate, but a kind of a different perspectives on issues, was a lady named Lynn Margulis, who was a very prominent biologist, a member of the National Academy of Sciences. And there were about 1,000 students in the audience. There was reporters from the New York Times. The president of the university was there. It was great. And after discussing it, I had a number of students come up saying exactly that they hadn't thought about it from this perspective before. They thought either that you had to be for evolution, quote unquote, or for creation, quote unquote, or that, you couldn't have scientific reasons for being skeptical of Darwin's theory. And they kind of scratch their heads and say, yeah, I'll have to think about that. And that's the moments that a teacher like myself just lives for.